Thank you, Dr. Brandoff, for that wonderful performance. The next segment of our program will highlight one of Dana-Farber's key priorities: precision, prevention, and early detection. To kick things off, and before our panel, you'll first hear from Carol Bowen. Carol is a strong advocate for prevention and early detection, so she is here today to share her experience in this area. Carol may be a familiar face to many of you, as this is her third year of attending the symposium. We're so grateful that she's willing to share her story with us today. Carol, I'll turn it over to you.
Thank you, Melanie, for that introduction. I'd like to thank Dana-Farber for the opportunity to speak to you about my experience as part of a cancer family and the importance of prevention and genetic analysis as tools for fighting cancer. During my life, I've come to view cancer almost as a family member, albeit one that I loathe. This unwanted relative barges in, unannounced and uninvited, into the lives of my loved ones and takes over, destroying plans and upending our lives. Each time it arrives, we're shocked, but helpless and powerless. I say this because 6 people in my family have died from pancreatic cancer alone, and other relatives have had, and in many cases died from, other types of cancer. The pancreatic and other cancers in my family have occurred in relatives on my mother's side. But if you knew my mom, you'd never believe this cancer was so rampant in her family. She's an amazing, healthy, vital 94-year-old who still walks 2 or 3 miles most days. However, her mother, my grandmother, 2 of her sisters, and 2 of her first cousins all died of pancreatic cancer. Other relatives have had breast cancer, cervical cancer, angiosarcoma, which is an exceedingly rare cancer of the lining of the veins, and one was recently diagnosed with lung cancer. So you can see why I say I'm from a cancer family. Pancreatic cancer is the only major cancer with a 5-year survival rate in the single digits at about just 8%. While the incidence and death rates for many other cancers are declining, those for pancreatic cancer are actually increasing. It's now the 3rd leading cause of cancer deaths, having surpassed breast cancer in 2016. Why is this the case? There's no single screening available, like a mammogram or a colonoscopy or a blood test. And because the pancreas is located deep within the abdomen in an area with few nerves, when symptoms finally occur, it's usually too late to operate, and as the disease has already metastasized to other organs. It's also particularly difficult to treat, as among other issues, the tumor typically forms a dense fibrotic shell around itself, preventing chemotherapy drugs from entering and destroying it. About 20 years ago, researchers at Dana-Farber and other institutions realized that early detection of pancreatic cancer might be more fruitful than trying to treat it once discovered. They completed research among people with several relatives who'd had pancreatic cancer and developed a prevention program aimed at discovering early stage tumors. This involves having an endoscopic ultrasound 1 year and a special MRI procedure the next year, and rotating these annually. Pancreatic cancer is rare under age 50, so when I reached my 50s, I decided to seek a proactive approach based on my family history. My mom had participated in the early CAPS, or cancer of pancreas screening studies at Johns Hopkins, which resulted in that prevention protocol that I mentioned. But I live in the Boston area, and researching online quickly led me to Dana-Farber as the place to go, and to the wonderful Dr. Sapna Singhal. She's the Director of Research at the Center for Cancer Genetics and Prevention. When I first met her, I felt an instant rapport. She was just what I was looking for in my quest to prevent pancreatic cancer, someone with extensive knowledge and interest in pancreatic cancer and cancer families, as well as a deep personal warmth. She answered all of my questions, and she spent as much time with me as I needed, and it was really refreshing. She clearly has deep respect for her patients and approaches them as colleagues, or maybe I should say fellow soldiers in this war. So now I have the appropriate procedures every year, and I'm fine, and I sure hope to stay that way. But unfortunately, pancreatic cancer did strike my own immediate family. My husband Les and I had long tried to take steps to prevent cancer in general, never smoking, exercising, eating a healthy diet, keeping our weight down, everything that you probably do too. By 2012, life was going well for the two of us. Our daughters were out of college, their careers launched, our businesses were doing well, and we were traveling and having fun. Early that year, Les had some stomach aches on and off, but nothing that seemed out of the ordinary. Finally, his doctor suggested an endoscopy to see what was going on. And it turned out that his stomach was just fine. But imagine our shock and our horror when, out of the blue, he was diagnosed with all things of stage 4 pancreatic cancer. He was truly the healthiest person I've ever known and a wonderful guy. Les received terrific treatment at Emerson Hospital in Concord, Massachusetts, and at Dana-Farber with the wonderful Dr. Peter Enzinger. And he got the latest extremely toxic chemotherapy, which he really tolerated well. He fought valiantly, but tragically, 4.5 months after being diagnosed and just a few months after his 60th birthday, Les died. After his death in 2012, I ran both his company and my own for a few years, and it was pretty crazy, as you can imagine. But in the quiet evenings, I thought long and hard about pancreatic cancer and what my daughters and I could do to help change the outcome of this horrific disease. As I mentioned, we were already seeking the support of Dana-Farber to help prevent cancer with early screening protocols. But I wanted to go further, and I concluded that developing even better prevention tools would offer the best hope for survival or even cure for members of my family and other individuals who are part of cancer families. And like all of you, I decided to support Dana-Farber, an extraordinary institution. I spoke with Sapna Singhal, and she told me that she and some colleagues were developing a research initiative focused on genetic susceptibility to pancreatic cancer called GENERATE, or Genetic Education, Risk Assessment, and Testing. I'm now funding her research looking at people who have a relative with pancreatic cancer that may be caused by an inherited gene mutation. Their study aims to teach these individuals about their risk of pancreatic cancer, educate them about genetic testing and its benefits, and then hopefully have them complete genetic testing through a simple blood test. Those who have a known gene mutation can be enrolled in protocols like the one I'm in with the goal of preventing pancreatic cancer. It's clear to me that genetics, early screening, and prevention are the future in the fight against cancer. Dana-Farber has long been a leader in this area, starting with the development of one of the first clinical genetics and prevention programs in the world from the seminal work of Dana-Farber physician, Dr. Frederick Lee. Remember when I said we felt helpless and powerless when cancer, that horrible family member, shows up one day? I'm supporting genetics and prevention research at Dana-Farber because I firmly believe the outcomes will give pancreatic and other cancer patients hope, and more importantly, a better possibility of survival. Someday, those of us in cancer families will be able to slam the door in cancer's face and walk away healthy and strong. I look forward to that day, as I know we all do. Thank you for letting me share my story, and thank you for your continued support of Dana-Farber.
Carol, thank you for sharing your extremely powerful story. We all want to slam the door on cancer. As Carol has eloquently shared, precision, prevention, and early detection is really about empowering individuals and families. So to continue this conversation, it is now my pleasure to introduce our three panelists. And following their introduction, I'll introduce the moderator for this live session. To begin with, Dr. Judy Garber. Dr. Garber is the founder and current director of Dana-Farber Center for Cancer Genetics and Prevention Program, one of the first clinical cancer genetics and prevention programs in the world. She also serves as the Susan F. Smith Chair at Dana-Farber. Next, Dr. Brian Wolpin. Dr. Wolpin serves as the director of the Hale Family Research Center, director of the Center for Gastrointestinal Oncology, director of the Lustgarten Foundation dedicated Pancreatic Cancer Laboratory, and is the Robert T. and Judith B. Hale Chair in Pancreatic Cancer at the Dana-Farber. Brian focuses his clinical practice on the care of patients with gastrointestinal cancers, with a particular focus on pancreatic cancer. And finally, Dr. Irene Ghobrial serves as the co-director of the Center for Prevention of Progression. She is a director of the Clinical Investigator Research Program, co-leader of the Blood Cancer Research Partnership, and director of the Michelle and Stephen Kirsch Laboratory. Dr. Ghobrial's work focuses on identifying what drives progression of precursor conditions in several blood cancers and how targeted therapies can be developed to stop these conditions before they evolve into cancer. I'm also pleased to share with you that Irene was recently named as one of the 3 newly established Lavine Family Chairs for Preventative Cancer Therapies as part of a transformational gift from Jonathan Lavine and his wife, Judy. Jonathan is a Dana-Farber Cancer Institute trustee. He's also co-managing partner at Bain Capital, and we are so pleased that he has joined us today to moderate this panel. It is my great honor to hand things off to Jonathan. Jonathan, good morning.
Good morning. Melanie, thank you for the introduction, and I want to thank Carol for sharing your personal journey and reminding us why we are all gathered here today, you know, for you and our many other patients. When I was asked to moderate this, I was at first a little surprised. My mother wanted me to go to medical school, but I didn't, and science has never been a forte. I know a little bit about cancer for 2 reasons. One is when you look in society today, besides the human toll it takes, the economic impact of cancer on our country is about 2% of our GDP. And think about just the burden that that's placing on families, on workers throughout the United States. So it's not just an emotional thing. Secondly, for many years, our family has had a privilege of a close relationship with Dana-Farber. That close relationship was established the hard way. Both my parents have been treated successfully at Dana-Farber dating back to 1995. And since then, we have felt a real obligation to give back. I've enjoyed serving on the board. My wife, Jeannie, and I, as I said, we've had families and numerous friends who have received advice and care from Dana-Farber's world-class physicians. We've seen the Institute flourish as a destination for families seeking the best possible care in the world. And we are proud that the Farber continues to extend their services in Massachusetts and beyond to ensure families from far and near have access to this amazing institution. I described once at a dinner that, you know, Dana-Farber is a miracle factory. Over the years, we have just been inspired by the level of excellence throughout the Institute. Its clinical care, the research that originates from its labs, the community programs, commitments, above all, to serve patients and families with compassion every single day. Dana-Farber is extraordinary, and therefore we are proud to have made the commitment that was described earlier to support Lori Blimscher's vision for efforts in prevention, early detection, and residual disease as part of the campaign for Dana-Farber. I know from talking with leaders here at Dana-Farber that cancer often begins to form years before symptoms appear. And for most cancers, the longer they remain undetected, the more opportunities they have to spread, making them much harder to treat. The key here is to be one step ahead, to outsmart and outmaneuver the cancer. Jeannie and I imagine a world where we could defeat cancer before it starts, supporting precision prevention, early detection, and elimination of minimal residual disease is critical to conquering and reducing the burden of cancer worldwide. Today, the promise of preventative care medicine for all patients depends on understanding an individual patient's unique biology so as to prevent and intercept the tumor before it can form and do any harm. Dana-Farber has the expertise to do this. The paradigm shift will be a game changer for patients. Dana-Farber is not only well positioned to lead the prevention and early detection movement, but also is impassioned to accept the challenge. Investigators in disease areas across the Institute are working to apply their expertise, translate discoveries into specific cancers, prevention, detection, and more broadly, strategies that are more applicable across many cancers. Dana-Farber's strengths in these areas are unrivaled, as is the culture of its team. I look forward to hearing the perspectives from some of the best physician scientists at the Garber and therefore in the world who have the knowledge and technology advancement to make significant progress. So I'm sure you'd like to hear more from me, but we thought since we have 3 world-class doctors and researchers on the panel, we had you hear from them a little bit. So I'm going to start with Judy. Cancer prevention is obviously not a new concept. For years, the public health sector has promoted the importance of altering modifiable risk factors we know can help reduce the risk of developing cancer, such as body weight, activity level, obviously avoiding smoking. Studies have also proven that reliable screening tools in early stage diagnoses can improve outcomes and reduce premature death. As a pioneer in the field of cancer genetics, can you begin by sharing with us how the field has evolved over the last 20 years and why now is the time to harness the power of advanced technology and research to develop comprehensive prevention, early detection models for all patients? Could you also touch on the similarities and differences between adult and pediatric care?
And only in a few minutes, of course.
Yes.
Right. So thank you, Jonathan, for that really thoughtful question. So I've been at Dana-Farber pretty much forever. I worked with Fred Lee, who was one of the earliest pioneers in cancer genetics. But when I went to David Livingston in 1990, when he was the physician-in-chief and said, you know, David, I think it's time for a clinic for individuals who do not yet have cancer or do not have cancer, who might still be cared for at Dana-Farber, David was all for it. And he found the resources from philanthropy to allow us to establish that first cancer genetics clinic, the cancer risk and prevention clinic. And it was also because at Dana-Farber, there was David, who was working on what BRCA1 and BRCA2, the first breast and ovarian cancer genes, did. Alan DeAndrea, who came from the pediatric side, but was working on DNA repair, and many of their trainees, that we could understand not only that there were these genes, but what these genes do, which makes it possible to think about targeting them. And in the early days, understanding that these were genes involved in repairing errors in the DNA, they figured out that there were whole pathways involved. And that let us find genes that conferred risk of breast and other cancers going down the pathway. So it wasn't just BRCA1 and 2, it was others as well. And that came from the laboratories. And treatments came too, because once you knew what the tumors, what these genes did, you could think about how to target their defects in the tumors. And then to recognize that it was not just breast cancer that was what BRCA1 and 2 in particular did, but also ovarian cancer, pancreatic cancer, and prostate cancer. And today we have studies that target treatments against all of these tumors, and even a new center where we're focusing on tumors that have become resistant in this setting. But now that I've been here so long, the children of my original genetics patients want to know, what can you do to prevent cancer in me? That's what we promised their parents. And that's where really the idea of precision prevention and prediction comes in, because we can have patients who start at a given age and start getting their early detection mammograms, MRIs, colonoscopies for our Lynch patients, a lot of burden of constantly looking over your shoulder. But we're not that good at trying to figure out within a family who's at risk when. How can we help people time the interventions? When do they have to start? When can they stop? When should they think about surgery? And what options do we really have besides surgery? So today we have 2 large prevention trials, one international study we're leading looking at a hormonal approach to prevent BRCA1 breast cancer. And it may not prevent, it may only delay, but that's okay, we'll take it. And with Miles Brown here looking at safe hormone replacement. We have to take ovaries from women at early ages. in Lee-Fraumeni syndrome, which was the first thing that Fred worked on, that links us to pediatrics because patients who have this condition get pediatric cancers as well as adult cancers. We've been working with the pediatric group on whole body MRI, a non-radiation tool for looking for early signs of cancers throughout the body. It doesn't sound as high-tech as we hope it will be, but at least is a beginning. And with Brian Crompton in the pediatric program, looking for circulating cell-free DNA, really early detection, which is what our patients really want. And when we find it, hopefully we can prevent those cancers from taking root, not just from needing treatment. These days, compared to the beginning, we have activity in GI cancers, including pancreas. We're still looking for genes for the families where we haven't found them, and it sounds like for Ms. Bowen's family, we haven't yet, so we can help them tell the difference between who's at risk and who isn't. Even in lung cancer, there are genes, and now we're working with the heme malignancies group to prevent people from having a bone marrow transplant with someone who has the same underlying issue that caused the cancer in the first place. And we're working with Dr. Ghobrial, so we get to work with everybody. Like you, we would like to see immune mechanisms exploited for prevention. That's a tall order. There's a lot of activity in treatment, but knowing what you're going to have to target, that's a challenge. And yet we have people willing to cross over from the therapeutic side to think with us. And the other place I think we really can look is to artificial intelligence, which will improve our interpretation of screening modalities, the radiology, and as well to help us with interpretation, even of pedigrees, who should be tested and perhaps who not. So we've had a lot of activity go on all these years, and I think that probably the message should be prevention is hard. You need the smartest people to work on it. That's why Dana-Farber has taken it on in such a big way.
Wow, thank you. Brian, pancreatic cancer is the third leading cause of cancer deaths in the U.S., with over 80% of the patients presenting an incurable disease, largely a consequence of diagnosis at a late stage, as Carol mentioned in her remarks. As a result, your team is focused on detecting the disease at the earliest possible stage. Could you share more about the studies your team is leading to detect, treat pancreatic cancer at its early stage and how it's developing?
Sure. Thank you for that question, Jonathan, and really do appreciate Carol's willingness to share her story. Pancreatic cancer is a difficult disease, and it's not easy to share stories like that. So I thought I would mention a couple different projects that we've been working on that get to Jonathan's point, and they also dovetail quite closely with what Judy was just talking about. So the first one originates going back 6 or so years ago, where we decided we were going to sequence tumors, so in patients who had pancreatic cancer, in an attempt to find new targeted therapies for our patients. And there aren't many for patients with pancreatic cancer. But as we were doing that, we also were sequencing the DNA that they inherited. And really, that was not done because we were specifically looking to find things that were inherited. We were really doing that only to try to inform us what was going on in the tumor itself. But as we started to do that program, even within the first 20 or so patients who volunteered and allowed us to sequence their tumor like this and their corresponding inherited DNA, we were repeatedly finding mutations in their DNA that they inherited, much more so than we expected to see. And that was surprising. And when we saw that result, which was not something that had been heavily described before, we went to go talk to who Carol had mentioned, Sapna Singhal, and also Matt Yergelin, who's another oncologist and geneticist in the group, and said, we're finding all of these mutations that we didn't expect in the patient's inherited DNA. Why don't we get together and try now to do this in a larger way? And so we were fortunate that many patients had agreed to provide samples to us over the years. And we went back and now sequenced the DNA that was inherited in hundreds and hundreds of patients with pancreatic cancer. And what we realized as we did that is approximately 10% of the patients actually carried a mutation that was inherited. And this was much higher than we expected to see. And because of that, we actually about 4 years ago at Dana-Farber changed our practice with Judy's help and Sapna's help, and started offering genetic testing for every patient with pancreatic cancer who comes to Dana-Farber. And we are quite a large center for Dana-Farber, so there's many, many hundreds of patients each year. So they would see, say, me in clinic as a medical oncologist and directly go to our genetic counseling team right after that. And if you look across these past several years, we again repeatedly see that about 10% of our patients carry an inherited mutation. And this is important, as Carol mentioned, both because of the implications for family and the ability then for family to get screening, and also because, as Judy mentioned, there are new therapies that are coming for patients who carry these mutations and get pancreatic cancer. So we then took this information, we compared it to a couple other large cancer centers in the US who we collaborate with. We then went to guidelines committees, many of which we sit on, and the national guidelines last year actually changed. So although we've been now doing this for quite a number of years at Dana-Farber, it is now considered a standard of care in the entire United States that every patient with pancreatic cancer should get genetic testing. And really that guideline traces back to some of the original work done here, and again at a couple other centers that we work closely with. So to answer Jonathan's point, it's really trying to say who's at risk for this disease so that we can start to do screening. So the second project I'll briefly mention is if this is maybe 10%, what about the other 90? Right? What about all these other patients who do not have a known genetic event that we can use to enter them onto a screening program like what Carol described? And this actually gets to another point that Judy made, which is this is the era of big data, right? Lots of data and machine learning, right? The ability to use computers to help us sort through large databases. And so what we've done is obtained a large grant from a group called Stand Up to Cancer to try to use large clinical databases to understand what are the patterns of symptoms and things that happen to people in the 1 to 2 years before their diagnosis. And as a medical oncologist who sees, unfortunately, every single week new patients with pancreatic cancer, even as a person and not a computer, I can tell there are clear patterns that occur in the year or two before. So how do we start to look in medical record systems to find those patterns and then alert doctors in advance that we think this person has a risk for pancreatic cancer in the next 1 to 2 years, and we should be doing something about that to find it early. And that's really the second large project I wanted to describe to you. That one we've been working on for about the past 18 or so months and making some good progress on. So I think to Jonathan's point, these are really just two of many, but two projects that are going on at Dana-Farber and with both the genetics group and others to find new ways to screen and identify pancreas cancers earlier when they're more curable.
Great, I appreciate it. And Irene, to you now. Although many patients with blood cancers are diagnosed with early indicators as precursor conditions, in most cases there are not any effective therapies available. This means that patients don't receive treatment until their cancer progresses. Can you talk about the steps that you were taking to better understand why some patients with these precursor conditions go on to develop progressive disease while others do not? And how are these findings being applied to targeted therapies to prevent progression?
Yeah, absolutely. Thank you, Jonathan. And I want to thank you specifically because of your help and because of your funding, I am a Lavine Family Chair for Preventative Cancer Therapy. And I think this is a big honor for me. So I'll take you back to what Judy said is why would we think of blood cancers and why would we want to prevent it? Just like solid tumors, every patient with blood cancers may have had a precursor condition, something that happens when they're asymptomatic, when they don't have any signs, and then the patients come to us when they have the symptoms, the back pain, the bone pain, the big lymph nodes. And blood cancers in general are actually well known to have precursor conditions that are clinically known. So we can actually diagnose people with something called MGUS, or monoclonal gammopathy of undetermined significance, or early CLL, even early lymphomas. And the new entity called CHIP, clonal hematopoiesis of indeterminate potential, that was actually described by Ben Ebert and many others here at Dana-Farber. And a few years ago, we said, well, when we see patients with multiple myeloma, which is the cancer that I treat, we actually tell them, wait until you have symptoms, wait until you have bone pain or fractures or anemia or kidney failure, and then I treat you. And it didn't make any sense to me. Why would we do that? In every other cancer, Judy was talking about early detection and prevention, we do not even do that in multiple myeloma. So we said, let's take a step back and let's actually truly prevention of multiple myeloma, but not only of myeloma, of all blood cancers. And we created the Center for Prevention of Progression. And this was really an effort with Ben Ebert, with Rob Seufer, with many others to say, instead of waiting for people to have symptoms, let's actually see them early, at the early stages of precursors. Let's detect what causes progression in them. And that's not only by clinical markers, but by genomic markers, like you heard, by also liquid biopsy markers, so we can detect in their blood samples what happens to them. And then we can also understand what are the immune regulators that lead to progression, and define the people who will truly progress in their lifetime and develop therapies for them. So we have a whole arm of therapeutic interventions or interceptions to prevent progression. And that was the beginning of the CPOC clinic, or Center for Prevention of Progression. And then we said, well, we don't want to just do that at Dana-Farber. We want to extend it to the United States and have the access to everyone who has MGUS and smoldering and CHIP mutations. And we created the first nationwide direct-to-patient access, something called P-Crowd. Any patient in the United States can give us samples, can give us clinical data. And with that, we have 3,000, 4,000 patients giving us information. And we were able to detect for the first time causes or mechanisms of disease progression from MGUS and smoldering myeloma to overt myeloma. And now we're taking some of those findings into the clinic to say who should be treated and who should not be treated when they have some of those genomic markers. And then a few years ago, thanks with Stand Up to Cancer, we started the first US screening study for blood cancers, specifically for multiple myeloma. And we said, if people are at such a high risk, if you know that 5% of the population, about age 50, will have MGUS, and we know that Black Americans have it at a 3 times higher chance, and we know that if you have a first-degree relative with myeloma, with lymphoma, you have a 2 to 3 times higher chance of developing MGUS, why wouldn't you be looking for it? And unlike mammograms or colonoscopy or endoscopy, you can detect myeloma and many other blood cancers by just a blood test. So we started the first US screening study called the PROMISE study, and that's screening 30,000 individuals in the United States now to try and understand who develops MGUS, why do they develop it, and then track or understand what causes progression in them and intercept it early, so that the hope truly is that we will never see a single patient with myeloma develop it when they have symptoms, when they come in with bone pain or renal failure. We detect it early and prevent it early in every patient.
Thank you. And Jeannie and I are very fortunate to be able to support you in your work, and are proud of putting our name on this incredibly important work that you do. Judy, back to you. You get the coronavirus question. There has to be. The pandemic has obviously impacted every facet of life, including essential cancer screenings, and the ways in which your team delivers care. Can you talk about how Dana-Farber Center for Genetics and Prevention, as well as the new Center for BRCA and related genes, has adapted to this time and continues to support those at the greatest risk?
Sure. I mean, obviously, coronavirus just affects everything. Here we all are doing this virtually instead of in person, which we all might prefer. You know, our cancer patients, my breast cancer patients, largely still come to the Institute. But genetics is something we can do virtually. And although we had thought about this before, what we're doing with coronavirus really made it now the standard, the way we do business. We see virtually all of our patients virtually. We see them by video, some of them by telephone. The genetic counselors meet with them. They already filled out an electronic questionnaire that lets us draw their pedigree or family tree. We work with the laboratories, which send them a kit that they can send a sample back just of saliva. They don't even have to get their blood drawn. And then, you know, we meet with them when the results come back. So we're able actually to make genetic testing available very broadly and very easily. And it allows us not only to take care of patients who don't have cancer and contact the Farber, but we're doing this increasingly for our cancer patients at Dana-Farber. So as Brian said, to make it easy for pancreatic patients who certainly have a lot else to worry about, to get their genetics done without making life more complicated. So I'm not sure how much we'll be going back to in-person genetics. Some we will, but this has really well, it has forced us, as innovation often comes, as part of necessity.
Thank you. And it's interesting, in all your comments, you've talked about working with others on this Zoom call as well as others in the Institute. Brian, could you talk about that collaboration a little bit and how that's progressing in the field of prevention and early detection? Are there specific examples of how you've been able to advance partnerships across Dana-Farber and in fact the entire Harvard system and beyond?
Yes. Yeah, thanks, Jonathan. Absolutely. I really think collaboration is key. I think, as Irene was saying, and Judy also, these are very complex problems, and they require really a team approach to solve them. And I think we've heard Dr. Glimcher and Dr. Hahn talk about tiger teams and bringing together groups of smart people to tackle very difficult problems. In pancreatic cancer, we do that every day. We sort of live that model through the Hale Center, which is our pancreatic cancer research center, where we bring together more than a dozen faculty from all over the Institute with different specialties to work together. And one of the main areas that we focus on in the Hale Center is early detection and prevention. So to provide a couple specific examples, I think we can go back to our prior discussion where we're talking about inherited risk in pancreatic cancer, right? And as we said, when we started that project, really that project was to understand the tumor and new therapies. It really did not have anything to do with trying to understand inherited risk. But once we started to identify these genes, we knew exactly who to go to, right? Because Dana-Farber has a tremendous legacy of expertise, as Judy has led and knows well. And there were obvious people for us to immediately go talk to. And then as we expanded the program, we did it in a way that's almost impossible anywhere else in the country to do, again, because of the team of people that are here. And then when we build on top of that, another really important collaboration that we've leveraged goes back to individuals who are studying, as Judy had said, some of these mutations in the laboratory. And we now have multiple clinical trials that are being led by James Cleary, who's a medical oncologist here, in collaboration with Alan DeAndrea, who Judy had mentioned before, and Jeff Shapiro, who have been studying in the laboratory how some of these genes work. And we now have a suite of clinical trials, actually 5 different clinical trials that are also actually funded by Stand Up to Cancer, to allow us to now try to help the patients we identify where we find these alterations. Again, these are not things that my own lab or my own group could handle by themselves. This really takes a whole team of people in order for us to study these diseases well and make a difference for patients. And then the last piece I'll add to that as you talk about beyond Dana-Farber. Yes, we have an amazing and incredibly deep expertise here, but we don't limit ourselves to only working with people at Dana-Farber. We absolutely collaborate across the country with others. And so I chair a steering committee at the National Cancer Institute for Pancreatic Cancer Early Detection. And this actually has 8 sites all over the country. And we work very closely now with a number of these sites to advance our research together, places like MD Anderson and Mayo Clinic. And again, we really are committed to bringing change to patients as quickly as we can. And that's going to happen when we work together, both within the Institute, but also beyond.
Great. And Irene, last question before we go to audience questions. And I think this sort of pulls it all together. When looking ahead for the next 5 years, where do you see the promise in the field of prevention and early detection?
Yeah, it's exciting. It's actually, I hope that we can see not only the next 5 years, but the next 10 and 15 years. I see that as a person who's getting older, and many of us are indeed getting older, I would love to see us have early detection, that I can get a blood sample and I can get to know not only my risk, genetic inheritance, but also whether I have an early potential cancer that I can detect, whether it's pancreatic, breast, or myeloma or lymphoma. Can I get it in a blood test? Can I get earlier interventions so that I do not have to wait until I have symptomatic disease and then get chemotherapy? My hope is truly early interventions, early screening, and early interception can change, like you said, not only the burden that we get as patients with cancer and the family burden that happens, but also the economic burden. And hopefully that will translate into everything. And this is happening. This is not something that's hard for us to believe, that we will get early detection methods with all of the next generation sequencing, with all of the artificial intelligence we're getting right now. I think that this is something that we will be able to get done. And I think the teams that we have here at Dana-Farber and indeed throughout the United States and throughout the world can make it happen.
Thank you. And we now have questions from the audience in this high tech. They're sending them in to me, so bear with me. The first question, I think we'll start with you, Judy. If a patient has a genetic basis for their cancer, how are they supported in, to inform other members of their family?
Great question. So for patients who have either been tested here or who see our team, we have genetic counselors, we have a psychologist in the program to help patients deal with the fact that, no one wants their family members to face the risk that they do. But if you find that there's risk, the goal is for you to help your family view this as an empowerment so that you can actually try to avoid the cancer that you're going through. So we try to help family members arrange genetic testing, which we can do here for many, but the laws don't let you test people in all states if you're not licensed there. So we help find people to arrange their testing, make testing easy for family members who may not be in Boston. But then we can still enroll them in trials and help counsel them about how to manage their risk, how to think about strategies for early detection, because there are standards for many, and for those that there aren't, to try to help design what makes sense, help them think about risk reduction, all of the things that they have to deal with. There are many things they can do today, but of course, this is Dana-Farber. We have to think about what they're going to be able to do tomorrow. And many of our families support the research. You heard about Sopna's Generate project. That's an example.
Got it. Thank you. And this is a question for all of you. So Brian, if I could ask you to start, can you speak to how big data and data collection and analysis is playing a role in research today and in your research?
Sure. So I think, as you probably can tell from some of the projects that we've been highlighting, data really has become key, right? So particularly for diseases that are not as common, right? So pancreatic cancer is the third leading cause of death from cancer in the United States, but it is much less common overall than other cancer types like lung cancer or breast cancer. And it's hard to study it at, say, one institution alone or in one clinic alone. And really, for us to get a handle on how to find these cancers early, we need large amounts of data. And so one of the projects that I was talking about before, where we're trying to use machine learning algorithms to look at patterns in patients before their diagnosis, benefits from having now millions and millions of patient records that we can then use in a de-identified way, of course, to try to understand what these patterns are before diagnosis. And that's really one of the projects we're working on. And that really has only become possible in the past, I'd say, couple years. If you go back 5, 10 years ago, a lot of the notes and the medicines we wrote were on paper, right? We were writing things down, or I would write it on a prescription pad and send it in. That's very hard to search and deal with. But when we have electronic medical records, where so much now is in data fields that can be searched and can be matched across health systems, it opens up a tremendous amount of possibility for how we can now start to integrate these data and use them for early diagnosis. And so I really do think that large data is a huge key to unlocking some of the potential that we have now for early detection.
Great. And Irene or Judy, do you have anything to add to that?
Data is everywhere. Artificial intelligence is part of what we all think about now, ways to make ourselves smarter using smart computers to help. So I agree with Brian, this is the future.
Right.
Yeah, absolutely. I think it's only when we have the large cohorts, when we have the 10,000, 20,000, 30,000 patient samples, then we can start asking the big questions that we just said. Then we can really make a difference into how can we change the future. We cannot do that with small numbers of samples, and we cannot do that with small number of data sets, whether it's samples or clinical data.
Great. And if I could ask you another question to weigh in on a question we just got from the audience, how does relapse prevention fit into progression prevention or slowing? What kind of work is being done there?
In the relapse setting or in prevention of?
And we're getting these live from the audience, so not exactly, but it's how does relapse prevention fit into progression prevention or slowing?
It's a great question. If you think about it, once we put patients into a complete remission or what we call minimal residual disease, and that could be lymphoma, myeloma, leukemia, or any other cancer, you're sort of resetting yourself. Am I cured or not? And if I'm not cured, what is my chance of progression? And can I prevent everything again? So it's the same work that we do in early prevention of progression from a precursor condition to myeloma is exactly the same idea of from a complete remission or from a potential MRD negative disease into a progression or a relapse. And how can I prevent it? What's the role of the immune system? What's the role of other cancer cells that are dormant, that are sitting there and we cannot detect them? How can we do better screening for those early relapses before it happens clinically? There are so many questions that we use exactly the same technologies and exactly the same data sets that we do in the earlier setting.
Great. And Brian, I want to direct a question we just received. If a family member were treated at Dana-Farber for pancreatic cancer, would you suggest other family members get genetic testing?
So that very much depends upon the family history and whether a mutation was identified in the family or not. And again, this really gets to the collaborative nature of things. So the patients that we see with pancreatic cancer are all seen by the genetic counseling team and physicians in Judy's group. And so these decisions are made in collaboration on an individual basis based on, again, the family history within that family, and also whether when the person who got pancreatic cancer was screened for a mutation, whether that person had a mutation identified or not. And I think there really is an important level of expertise that is required for this, right? Again, it's not something that every small center around the country can do. You really need a lot of experience with these families to know how best to counsel them on who should be screened and how to do that.
Jonathan, though, if someone's family member was treated in the days before we did genetic testing on everyone, then they could still consider genetic testing. They could participate in the GENERATE trial that Carol kindly mentioned that's Opnacin running. Or they can see their physicians otherwise. So even if someone was treated longer ago, the family may still decide that genetics is important for them.
Appreciate that addition. And actually, can we start with you? Perhaps you could share a brief patient or family story that continues to inspire your work, or remark how stories like Carol's inspire your work. And this will go to all of you, but Judy, why don't you start?
Well, so, Fred Lee worked on the Lee-Framini syndrome. He never wanted to have his name on this, but it was an important condition that's caused by a gene called TP53. So Fred first described this in the 1960s and found the gene here at Dana-Farber in 1990. And just the first 2 weekends of October, we had a conference, all virtual, all day, Saturday, Sunday, both days of the weekend, with scientists from around the world and families with Lee-Framini syndrome, which is not that common, fortunately, a condition. And I have to say that the opportunity to work together with scientists and families all at once really keeps you grounded, because the families know what the important questions are for them. Here are people who have a risk for many different kinds of cancers, including childhood cancers. And they really want to know, what are we doing to try to understand how to treat their cancers, but more how to find their cancers early and prevent them. And TP53 is a tough gene. It's the gene most frequently mutated in tumors. Fortunately, not the gene most frequently mutated in people, because it's hard to target and it's hard to use, but it has been studied and continues to be studied. And every time we get together like this, we come up with many more questions, many more projects to do, and a group of people who, they're patient, but not really. They want answers and they deserve them. We have to work harder.
Okay, Brian?
Yeah, sure. So I think, as Carol's story demonstrates, there's a lot of bravery and amazing people who go through pancreatic cancer, both as a patient and as families. I think a patient who illustrates some of the concepts we're talking about is a patient I take care of in clinic. And he actually was one of the first patients that enrolled to that tumor sequencing protocol I've now mentioned a couple times before. And he was one of the first patients we identified that had an inherited BRCA2 mutation as part of that study. Turned out there were multiple more after him, which is what led to some of the work that was described. So with his pancreatic cancer, once we knew that that mutation had been inherited, we made sure he received a type of chemotherapy that was known in other types of cancer, platinum-based therapy, that would be effective in someone who had a BRCA mutation that was inherited and then influencing the biology of their cancer. And he's now, because of that and some amazing work by our surgeons, is now cancer-free over 4 years later from pancreatic cancer, which is not so common, but was a fantastic thing that resulted from knowing that information. His family, he has 2 daughters, then were seen in Judy's group. And when they saw the 2 daughters, offered them genetic testing, which they both agreed to do. And one had the BRCA2 mutation and one did not. So one knows that the risk of these cancers that include pancreas cancer is not as relevant to her because she does not carry that mutation. And the one who did actually came, this was right before COVID, came to clinic with her father. And, I was chatting a bit with them, and she said she was getting an MRI, but she was only in her 30s. So I said, oh, what kind of MRI are you getting? And she said, a breast MRI, right? And this is because when we find individuals who have these mutations that run through families, it's not only relevant to pancreas cancer or to a single cancer type. This is really relevant to prevention and early detection of many cancer types. And so we can see how now the information gained in the patient that I took care of with pancreatic cancer gets passed down to other generations in the family. And we start to now make a dent in overall incidence of cancer and preventing cancer and finding it early. And I think it, again, is just a very gratifying way to see that the research that goes on at Dana-Farber then influences the patient in front of us, but there's a whole cascade effect of other patients that it then helps. And ultimately, I think, as we've all mentioned, tries to put this to rest, right? So we find cancers early or prevent them altogether and prevent so many of these deaths from pancreas cancer and other types.
Irene?
Yeah, I think about every clinic that I have, every single patient gives me a story that is unique and is special. And when we started the PROMISE study, I didn't think of how much impact it will have. And when we're screening people who would not have known that they may have myeloma, and for the first time, you actually not only diagnose an early precursor, but you may actually diagnose myeloma that was about to happen. And one of those stories is a 41-year-old young woman who screened on the PROMISE study because her sister died of myeloma and her father died of myeloma. So it's really a family history. And she's Black American, she had no insurance, and we screened her and we were able to find indeed a monoclonal protein that's high enough that she has almost a very high risk of developing myeloma in the next few years. And it was really an effort that we worked through, making sure that we can get insurance, making sure that we can get care for her, and getting early prevention to potentially prevent the same effects that happened in the same family and potentially treat early and have a difference in their survival. So I truly think that we should not be waiting. What are we waiting for to treat our patients? Early detection can really save lives in our patients.
Thank you. So Judy, Brian, and Irene, thank you for joining us today and for your incredible work. Dana-Farber is an institution. When I walked in there with my mother to start her treatment with Dr. Friedman when I was 29 years old, it terrified me. And in the intervening years, it has become an institution that inspires me. And one of the things that is so clear, that an institution that does such amazing work at treating and discovering therapies is the place that will help prevent and come up with cutting edge early detection. Because every single patient, when they walk in, wonders, if I had just found this earlier, if I had just seen this earlier, and their family, should I have made them go to the doctor? And, Dana's work in this area is designed to benefit patients along the entire continuum of cancer care, from those who are healthy to those living with precancerous conditions, as well as patients, treated for cancer who had a risk for recurrence, and those living with cancer metastasis. And listening to you here today reminds me why it is that Dana-Farber is so inspirational, and why we're so proud to support your work, to join so many others who support the work of this Miracle Factory. And I thank you very, very much for all that you do, for all the people whose lives you save and change, as well as for taking the time with us today to share some of your stories. Now I will turn it back over to Melanie, who will provide remarks before lunch.
Thank you.
Thank you, Jonathan, Judy, Brian, and Irene. One of the silver linings of this remarkable time is that we probably never would have been able to have all of you in the same room, because you're all so sought after in your fields. And Jonathan, thank you so much for your personal remarks and for navigating the live questions. It was a really, really terrific conversation, and it provided an excellent overview of the projects and the innovative approaches taking place at Dana-Farber every day. There is such tremendous potential in this area, and you can all understand why this is one of Dr. Glimcher's strategic priorities. We are all committed to reducing the burdens of cancer for generations to come, and are so grateful for the philanthropic support of so many of our attendees who have joined us for the symposium today. So thank you, and thank you for your many questions throughout the afternoon or the morning so far. As Irene said, with work like the PROMISE study, the hope is that we never have a patient who truly develops multiple myeloma thanks to early interventions. And statements like this exemplify the power of our early detection and prevention efforts. Equally powerful is hearing themes such as how the standards of care in the US trace back to work conducted by Dana-Farber faculty and at Dana-Farber. To all of you, we appreciate your active attention and questions, and you're tuning in from so many different places around the city and around the country. So before we break for lunch, I want to offer a few housekeeping reminders and suggestions. We'll be breaking for lunch for an hour, and at that time, at 1 o'clock, we're going to offer 7 different breakout sessions. So actually a little less than an hour. We will always we've always done this and in person, and we're going to continue to do that tradition even in our virtual environment. I'd now like to take a moment to thank the leaders of each of these sessions and provide an overview of the options for all of you. We'll have Discovery Science, which is led by faculty co-chairs Dr. Bill Hahn and Dr. Kevin Haggis, and the trustee co-chairs trustee co-chair Dr. Harvey Berger. Our Gastrointestinal Oncology breakout session will be led by Brian Wolpin, who you just heard from, and the trustee co-chair is Dr. Winnie Wong. Our Hematologic Oncology breakout session will be led by faculty co-chairs Dr. Margaret Shipp, Dr. Robert Seufer, and trustee co-chairs Mark Cohen and Ted Passarello. Our Pediatric Oncology breakout will be led by co-chairs Dr. Scott Armstrong and Dr. Lisa Diller, and our trustee co-chairs Allison Pourvue-Jaffe and Conrad Wetterow. The Lowe Center for Thoracic Oncology will be led by faculty co-chair Dr. Pasi Jani and trustee co-chairs Alice Cutler and Bill Gillen. And then the Susan F. Smith Center for Women's Cancers will be led by our faculty co-chairs Dr. Alan DeAndrea, Dr. Judy Garber, Dr. Ursula Matalonis, and Dr. Eric Weiner, and our trustee co-chairs Jane Brock Wilson and Jane Jameson. And finally, Treating Cancer Across the Ages, Children to Adults, will be led by faculty co-chairs Dr. Steve Sallin and Dr. Deb Schrag, and our trustee co-chairs Nancy Gibson and Jen Perini. We welcome you to scan below and look on the individual sessions to see a more complete agenda for each planned breakout session. And for those of you who attend the symposium in past years, We know the materials that we have handed out during the event have been a helpful resource for you. We've included much of that same content in the Resources tab, which you can find at the top of your screen. These resources will continue to be available within this event platform throughout the year ahead, and we are happy to have follow-up conversations specific and tailored to your areas of interest. So thank you very much for your time and attention this morning. It has been a great showcase and conversation, and please note that tomorrow you'll receive an email message from Dr. Lori Glemsher, which will provide a quick 3-question survey. Your feedback matters to us now more than ever, and we hope that it will continue to shape our future events. So at this time, I'm going to ask you to take a break and enjoy your lunch. And at 1 o'clock, we'll welcome everyone back for our breakout sessions. You are welcome to join the breakout session of your choice. And at this time, we're going to ask that you click out of this window and scroll down to the breakouts available below, and then click within that session to see the content. The video feeds are located below the agendas. And we'll end the afternoon at 3 o'clock with final remarks and thanks from Dr. Lori Glemsher. So again, thank you very much for your attention. Thank you for joining us. There are hundreds of you on this call today and attending the symposium virtually. And I want to thank you, and I want to thank the staff who has worked diligently over these many months to put this program together. And again, without our faculty, we would be in a completely different conversation. We're so grateful to all of them for taking the time to participate today. Enjoy your lunch. Bye-bye.